All right. So first, thank you to the organizers, and then also thank you to all of you who decided to show up here at 8 a.m. on a Friday morning. So if you could tell from my long title back there, today I'm going to be talking about chromosomal inversions. But before I do that, I'm going to give a very broad but brief overview on the evolution of genome structure before I get into some of the specific hypotheses that we're testing with regards to our system. So between species, we can have differences in the total number of chromosomes. So for instance, some plant species can have chromosome numbers in the hundreds, whereas other organisms, such as the honeybee, might have far fewer. Between species, we can also have differences in the orientation and arrangement of the individual homologous chromosomes. So it's clear that the structure of the genome is fluid. However, the evolutionary mechanisms that establish these large-scale rearrangements remain unclear. It's possible that the evolution of complex traits may be driving how genes are distributed and ordered along a genome in order to control how often new combinations of alleles are generated through the processes of independent assortment and homologous recombination. So recombination, of course, results from crossing over during meiosis, where its primary purpose is to ensure the proper segregation of chromosomes. However, it's also a mechanism for shuffling genetic variation and providing new combinations of alleles for selection to potentially act on. So there's both theoretical and experimental evidence that has demonstrated that natural selection can be more efficient and adaptation can be more effective in the presence of recombination. However, there's also this effect of recombination that it can break apart favorable associations between beneficial alleles. And in the case of local adaptation, it can exchange potentially maladaptive migrant alleles onto a chromosome. So there appears to be this battle between generating new combinations of alleles versus keeping those favorable associations together. So a type of structural variation that can modify this level of recombination are rearrangements or inversions. So inversions occur when there's two double-stranded breakpoints and the resulting fragment of chromosome gets reinserted end-to-end in reverse orientation. So inversions have generated a number of fixed rearrangements between species. So for instance, between humans and chimps, indicated by these red lines here. And of course, I'm in a Drosophila section, so I probably don't need to remind most of you here that there's a number of fixed rearrangements across the chromosome arms of the Drosophila genus. But besides existing as fixed differences between species, inversions can also be polymorphic within a species. So between two different chromosome arrangements, you get the formation of these inversion loops. And so in these inverted regions, crossing over and therefore the rate of recombination is drastically reduced. There's evidence in a variety of different systems that inversions might be adaptive or actually selected for in natural populations. In the specific case of the third chromosome of pseudoobscura, which is what I'll be focusing the rest of my talk on today, there's evidence that it's adaptive because the relative frequencies of these arrangements are clinally distributed along the American Southwest. So moving from west to east, particular arrangement frequencies might increase while others decrease. And this clinal distribution has been stable since it was initially described nearly 80 years ago. So despite their overwhelming ubiquity in nature, we don't have a clear understanding of the evolutionary mechanisms that can establish inversions within natural populations. There's a number of different hypotheses, however, that are used to suggest how this process might occur. So the first hypothesis is that inversions could be established through their direct physical effects. So this is where the breakpoints might physically alter a protein coding region of a gene or modify its regulatory elements. And in fact, for example, in many human polymorphic inversions, this is the case. A second hypothesis is that inversions might hitchhike along with an adaptive allele. So this is where there's a single beneficial mutation sweeping through the population, and inversion happens to capture it, and it gets brought along with it to high frequency. A third hypothesis is that inversions are established through genetic drift. And so there's been a number of theoretical models that have been developed that predict that in smaller effective population sizes, there's an increased probability of an inversion become fixated. And the fourth class of hypotheses are that inversions are established through their indirect effects of suppressed recombination. And so we've shown previously that on the third chromosome, as expected, in regions where multiple inversions overlap, we get a significant suppression of recombination, as indicated on the y-axis here. And this could be beneficial for a number of non-mutually exclusive reasons. So for instance, it could capture a segment of chromosome that's relatively free of recessive deleterious mutations. It could be holding together multiple loci that interact beneficially, so a form of epistatic selection, or it could capture sets of locally adapted genes or alleles. So in our work, we're taking an integrated approach where we sequence both the genome and transcriptome of different third chromosome gene arrangements to test some of the predictions of these hypotheses in order to gain an insight into the evolutionary mechanisms that can both establish and maintain inversions within natural populations. So we sequenced the full genomes of 54 isochromosomal males, and these are representative of 6 different arrangement types. And for these same arrangements, we also tested for differential gene expression across 3 different life stages in larvae, females, and in males. So each third chromosome gene arrangement is named after the location in which it was originally discovered. So for instance, CU for Cuernavaca or PP for Pikes Peak. And so what this diagram is depicting is how the different syntenic regions of the third chromosome have been rearranged because of this series of overlapping inversion mutations. And so with the exception of the hypothetical arrangement, all of these are still extant in nature. And because all the inversions are overlapping, by analyzing the ordering of these syntenic regions, we can determine the evolutionary relationships or the phylogenetic relationships between these different arrangements. So we can determine which chromosome was derived from which ancestral arrangement. Previous work has shown that the arrowhead arrangement, or AR, is the youngest within our sample. However, in nature, it's also the most widespread. So this would seem to contradict the predictions of the hypotheses that these inversions are established through drift, because under these models, the frequencies of an arrangement would be directly related with its age. We mapped the locations of each of those inversion breakpoints using large insert Illumina mate pair sequencing. And in order to find the breakpoints along the chromosome, we searched for regions such as this example, which is shown here. These are regions where the reads are mapping at greater distances than their expected insert size and in reverse orientation. And so after mapping all of these breakpoints, in no case did we find evidence that the breakpoints themselves physically disrupted a protein coding gene. We did find one case in the gene expression data where the standard arrangement had greater levels of expression than Pikes Peak, just in larvae of a single gene. But with the exception of this case, in our data we find limited evidence to support the hypothesis that these inversions are established through their direct physical effects. We also searched for signals of adaptive evolution among the genes along the third chromosome to test the predictions of some of these other hypotheses. And so we developed a method which we call the population-specific branch length. That's very similar in nature to previously developed methods, but it's testing for genes that contain significantly high proportions of arrangement-specific allele frequency changes. Now, a strength of our data set that allowed us to calculate this statistic in the way that we did is that, as I mentioned, we can infer what the relationship is among the arrangements based on the ordering of those syntenic regions. And so for each gene, we can fit our data to this inferred phylogeny in order to estimate pairwise genetic distances and therefore build our branch lengths. So what we can see is that we get an overall general increase in the length of branches within the inverted segments of the chromosome, indicated by these dashed vertical lines. We also get a greater proportion of significantly long branch lengths within these regions. And this is true for nearly every arrangement within our data set. And so we see evidence that there's multiple loci showing evidence of adaptive evolution that are being maintained within the inverted regions. So this seems to provide limited evidence to support the hitchhiking hypothesis. So among those genes that are showing evidence of adaptive evolution, we also estimated linkage disequilibrium, or LD, in order to measure the strength of association among those sites. And so what we can see is we get extensive LD generated within the inverted segments of the chromosome. We also see significant long-range associations. However, these are also broken up by blocks of random polymorphism, and so we see evidence of gene flux. So the inverted regions are not holding together and maintaining complete blocks of linkage across the chromosome. Instead, we get that they're maintaining significant long-range associations among those sites within genes showing evidence of adaptive evolution, but these are also broken up by random blocks of polymorphism. And these seem to support the predictions of hypothesis number 4, that these inversions are established through their indirect effects of suppressed recombination. We tested for differential gene expression, as I mentioned, across 3 different life stages. And so first, we get a general pattern that when we just use the patterns of significant differential gene expression, when we perform an unsupervised hierarchical clustering, like what is depicted up at top, for each life stage, we see that this generally recovers that inferred cytogenetic phylogeny. So the pattern of gene expression is related to the history of the chromosome arrangements. For genes that are showing evidence of significant differential gene expression, we see that they're enriched in both odorant and taste perception, so sensory perception. So we see multiple transcriptional differences maintained by the inversions. And so again, this provides limited evidence to support that this is just a single adaptive locus that was captured by the inversions. So in addition to the population-specific branch length, or PSBL analysis, we also search for signals of adaptive evolution by looking at high mean-derived allele frequencies, skewed allele frequency spectra using Tajima's D statistic, and unusually long haplotypes using the IHS statistic. And so for each gene arrangement, we came up with a set of genes that are showing evidence of adaptive evolution by requiring that they tested significant for at least one of these common tests, and also contained at least one fixed amino acid difference unique to that arrangement, or showed evidence of differential gene expression. And so there have been previous theoretical models that have predicted that the size of an inversion is actually a selected trait, so that if inversions are established through their indirect effects of suppressed recombination, that the number of genes underlying local adaptation or epistasis may be directly related with the size of the inversion. And what we see in our data is that within each arrangement, the proportion of genes that are showing evidence of adaptive evolution, we see that this is directly, linearly, and positively related with the size of the inversion. So we see empirical support for some of those theoretical predictions. And we do not see the same significant relationship in the uninverted proximal or distal regions of the chromosome. So for genes showing evidence of adaptive evolution, we also performed a gene ontology or GO analysis to look for common enrichments of biological functions or protein domains. Again, we see an enrichment of odorant binding, of pheromone binding, so genes that seem to be involved with sensory perception. So this could support a model of local adaptation across a heterogeneous environment or epistasis through the indirect effects of suppressed recombination. However, what I just want to end my talk on is a potentially intriguing result that we see with genes enriched in the limonene and pinene degradation pathway. So limonene and pinene are monoterpenes or chemicals that are produced by a variety of different plant species. And in nature, they act as antifungals and as insect repellents, particularly for pine beetles. What makes this result interesting is that work in the 70s demonstrated that within the ponderosa pine, the relative composition of these chemicals varied clinally along the American Southwest. And so in this figure, we can see the relative compositions indicated by the red and green bars. We can see that there's substantial variation across the American Southwest. And this same clinal distribution overlaps with the same species range as pseudoobscura, so with that same clinal distribution of arrangement frequencies of the third chromosome. And so perhaps the arrangement frequencies are directly or indirectly related to this clinal distribution of limonene and pinene. And so although, right, as it stands now, this is nothing more than a suggestion, I think that this provides a potentially intriguing result. So despite the wealth of genetic knowledge that pseudoobscura has provided us over the last 80 or so years worth of research, we still know relatively little about its life history or ecology and nature. So I think this provides an intriguing result that perhaps we may be able to infer something about an organism or a species ecology just based on its genetic data. And so with that, I would just like to thank my advisor, Steve Schaefer, the rest of my committee members back at Penn State. I guess these are all now former members of our lab, as some of our undergrads have recently graduated, our main collaborator at Baylor, and my sources of funding. And so with that, I will take any questions if there's time.
Time for a couple questions. Anyone? I have a question. Is it known how inversions might change the 3D conformation of DNA? Is that a factor in this at all?
So we haven't looked at that at all within our data. There is some preliminary work that has been done out there, but with regards to our data, we don't have any information about that.
Any evidence of length variation?
Of length variation?
Length variation. There's no a priori reason there shouldn't also be length variation.
Yeah, so I get, like, do you mean linked variation just across the chromosome within each arrangement? Yeah, so we do see evidence of linked variation. So I guess I can show here. So this is, so I showed earlier a plot of LD just using the genes that are showing evidence of adaptive evolution. This is a plot of LD using every segregating site across the chromosome. And so we do see evidence of linked variation that's generated through the inversions.
Okay, thank you.